Hey all, welcome to ShareTrack. This is Raj here. As a non-technical investor, I find it difficult to make sense of genomics, but it's a very tempting field to invest in. And since I believe that we need to know what we are investing in, I invest my time trying to understand the topics. So in the process, I put a lot of hours and go through a lot of publications and everything. And to the best of my abilities, I try to gather sufficient information. And that's why I have this channel where I want to share all those information with you. And it would do well for you to do further research and make sure that you have a good understanding. I'm not a geneticist or a doctor or a scientist, but I'm coming at it from an investor perspective. And today I'm going to share with you a new innovation that has happened at Broad Institute. And it's very exciting because it tackles the issue of delivering the therapy into the cells. So let's get started. A quick recap. There are three methods of performing gene therapy. The first method is to replace the defective gene. The second method is to add a gene to perform the work of the defective gene. And the third one is to turn off the defective gene. And using these three methods, scientists go about creating new therapies. Well, so far we have been aware of the fact that delivering the therapy to the right cells in the right amount has been a problem. Also, they have to deliver it to the right cell. And if it's not delivered to the right cell, then off-base edits can happen. These are all the known risks with the therapy. And these are the factors which potentially delay a therapy from advancing in various stages in the FDA approval process because it reduces the efficacy and it increases the risk of delivering a therapy. And therefore, any innovation that comes in the field of delivering gene therapy into the body is most welcome. Generally, when in vitro applications are done, it's much easier because it's outside the body, it's in a petri dish. But once you introduce the gene therapy into the body, a whole lot of new things starts clicking in. There's the immune system that starts kicking into gear. And of course, the whole body is available. So if you were to deliver gene therapy through virus, then the first thing that they do is remove all the malicious content of the virus and then put the gene therapy that they want to deliver into the body. Now, this virus can go anywhere in the body, and therefore the chances of off-base delivery and off-base edits are quite high. So there has been experiment of late of creating virus-like particles, and the approach taken by Broad Institute is similar. But what they have done is they kept on working on improving the virus-like particles the same way that we do in software development. So you first develop your base version, and then you run a test on it, and then start fixing the defects, and then run a test again, start fixing other defects. So you keep on putting patches and releases until the whole software system becomes absolutely perfect, and all the defects and validations, everything is in place. Something similar was done by Broad Institute team with their virus-like particles. They started off and they kept on making modifications and engineering the virus-like particle so that it reached a stage where they were able to deploy it both on animal and human cells, and they were able to be very, very precise in the delivery, and they were able to deliver the right quantity to the right cells. They have been able to do all those things. They tested it out in mice and it worked out well. So they have restored partial vision in mice. The finding they had was that their virus-like particle mechanism is much more suited for delivering proteins as part of therapy, proteins delivered to specific cells, specific proteins to specific cells. And as a result of which they have a much higher efficiency rating. However, this method is not suitable for delivering DNA therapy. You can deliver a protein, but not DNA. But Cas9 is also a protein, so it works well in that manner. So this particular innovation is really good. And another thing that stuck from stuck to my mind from reading their article is that the team feels that the approach that they had taken of progressively improving the virus-like particle by identifying issues and then making changes in order to overcome the issue, that approach can be used for delivering other payloads also. So it's just a question of going through a methodology of constantly improving the virus-like particle. That's my understanding as a layperson. But this is definitely a very exciting development, and this can be used for both base editing as well as performing editing using CRISPR-Cas9. And I think this is going to help many companies to improve their therapy design as well as efficiency with which they are able to do it. So that said, this is just a brief video to bring this new development to you. If you like this video, please press a like out there. And if you have not yet subscribed, please subscribe for everything related to CRISPR and genomics talks. Thanks and have a great day. Bye for now.